accurate. It probably doesn't take into account native people, it takes into account pollinators. So, okay, so here's our lock operon. So this is where we left off last class, defining operons, defining the parts of an operon. I would, hope that you're becoming more familiar with these types of questions and these diagrams that really help us to think about mechanism. That's why here's the book. So just to everyone get on the same page, how is the lock operon being regulated? So this is our first pop-back question. Under conditions that we've talked about. So there's a big clue in this figure. So what are people paying attention to when they're answering this question? Currently we have an even split for ABC, with about 60 people answered. So what are you guys picking up on to try to identify the regulation of the operon?
Repressor.
Any thoughts? What are all of our options? We can have So we literally, I don't, you guys can't see the distributions on top of that, right? It's an even split. People have answered A, B, or C at equal frequencies. We have two possibilities, and we combine some of these words together to make really specific comments about this operon. So what do you guys want to choose? Choose your own story, positive or negative. And why? Yeah, this pencil's not working.
I chose negative because the protein is a repressor.
Yeah.
Inhibiting transcription.
Yeah, so, well, I'm too distracted by this pencil not working. I agree with the first part of what you said.
Okay.
Let me try to close it so we can do it. It's going to be very difficult to do this if I can't have a pencil. Okay, I'll try to do my best with my finger, and it'll be really illegible. We'll see. Negative. There's a regulator that's binding to the promoter, right? So when we have a choice of positive or negative, it's really from the perspective of the operon, what's interacting with it, and how it's interacting with it. So it's interacting with negative men. go negative due to this. So that's the big clue. Number one, in the absence of lactose, the regulator, the repressor, is binding to the operator. So now we have inducible repressible. Right? This is where I stopped paying attention, unfortunately, for the second.
Well, it says binds to the operator and inhibits transcription, so inhibiting transcription makes it a negative as well.
Yes. So what about inducible versus repressible?
You look at the initial state, if the repressor is bound or not bound.
Yeah, so you can look at the initial state of the regulator or the initial state of transcription. So if something's off, then on, it's inducible. If something's on, then off, it's repressible. So, and we're usually, the before and after is dictated by either the co-inducer or the co-repressor, the small molecule. So in the absence of lactose, again, that's not the inducer, it's allolactose because there's metabolism that happens. But that's how we often judge this system. So this is the before picture. So before it's off, so that's going to give us inducible. Right, so together this is, oh my gosh, this is the worst.
He's stuck on his pen, not working.
So this would be negative inducible. So the best answer here is negative inducible for the reasons we just gave. There's a repressor that's binding to inhibit transcription, so transcription is off. Then when lactose comes, it inactivates that repressor and transcription will turn off. I apologize, this is really bothering me. Give me 1 second, because otherwise I cannot do this efficiently. That's fine, but it's more important to think about the logic that we went through rather than whether you got the answer right. That's just about participation. Okay, so here's the after, the presence of lactose. So that's how we're measuring this system. But again, it's not lactose that's the inducer, it's allolactose. There's metabolism through that beta-galactosidase enzyme. It binds physically to the regulator and inactivates it. So we had before transcription turned off, and now transcription's on. So that gives us an inducible before after. So our algorithm for thinking about operators is what is the conformation of the regulator in the absence of any small molecule? Is it binding or not? And then how does that relate to transcription? Is it off and on? So now we're going to deal with mutations. So we have this system, we sort of understand it in the sense of what's happening in the wild type condition. Now let's introduce some mutations to think about their consequences on gene regulation. So a couple terminology things when you're reading these mutations. So if we wrote the genotype for a normal bacterial cell, it's haploid, right? Bacteria only have one circular chromosome most of the time. So if we wrote a wild type lac operon, it would look something like this. We'd have lac I, we have that. It's named inducer because we now know it's inducible, but this is the regulatory gene. What are the other parts of the operon? So lac-I is not in the operon, but it's a really important aspect of the genotype of this system. So what are our other pieces of an operon?
A, Z, and Y. Like A, like Z, like Y?
Sure, structural genes, yeah. I'll add those in a second. What else? So it has kind of a certain orientation. So usually you write genotype from left to right in these systems. It doesn't always have to be the case. It doesn't always matter, but bacterial do have polarities to their chromosomes. So we need a promoter on that operon, right? promoter, we have our operator, then we have our structural genes, like Z, like Y, like A, like that. We usually don't think much about like A in these problems, because if something's happening upstream, like A is going to be the last thing to be expressed anyway, because it does have that order. So that's why order matters. So remember, like I is not part of the operator, but it's a really important part of the system. But this is all the operon. So if we study mutations just in this case, we kind of be limited in what we can do, because everything's kind of in complete linkage, right? Bacteria only undergo recombination, and frequently it's very different than kind of eukaryotic recombination. To make a system that you can actually disentangle things, you need to separate components. You need to be able to separate different promoters from different structural genes, from different regulator genes. So in bacteria, you do that by making what's called partial diploids. So diploid has two copies, right? In bacteria, you don't have two full chromosomes. You actually have kind of the endogenous bacterial chromosome and then a plasmid. So a plasmid is a small circular piece of DNA. Some bacteria take them up. They might have other genes on them. They're pretty common, but we can use them for our purposes in creating systems where we can do genetics. So what would that mean? We can put parts of this operon on a plasmid, and now we have what's called a partial diploid. It's not diploid for the whole genome, it's not diploid for the whole operon, it's diploid for whatever genes we put on that plasmid. So if we put just structural genes on this plasmid, we could write the genotype like a fully diploid genotype. So the slash is now referring to genes on a different chromosome, right? So the two kind of complement. So in the bacterial chromosome, we might have a LOCK-Y mutant, but then we're going to kind of complement this with our plasmid. So you can either write pluses to identify wild type, or the absence of a letter usually identifies wild type. What you really want to pay attention to are those superscripts, so minus. We'll see some other letters that are very specific to things in the operator or the regulator. But this is how you would write a diploid, a partial diploid. And again, it's showing linkage. The big slash is denoting kind of anything to the left is on one chromosome, anything to the right is on another chromosome, but they can still interact if they have the capacity to interact in kind of what we call strands. Any questions on this?
What's this slash through the L? On the Y one, is that a mistake?
So we're going to do some top hat questions where we'll write a genotype that I'm going to ask you to think about the phenotype. So where can we see mutation? Anywhere. And they'll have different consequences, especially in these partial diploid systems. So the first mutations we're going to think about are not in the operon, but they're in that regulator gene. So we can think of kind of a LOCK-I minus mutation. So this is a non-functional repressor. So in a, in a haploid system, what might this look like? So what is the job of the repressor? So this is not in top hat. We're going to do partial diploid in top hat. Just in a normal scenario, a haploid scenario, what's going to happen due to this mutation in the repressor?
Is that going to turn it on?
So what's the job of the repressor?
Doesn't it block it?
Yeah, so it's repressing transcription, turning transcription off. So if it's a mutant, if it can no longer do that, what's going to happen?
It's going to continue to transcribe.
Yeah, so I would say constitutive. So constitutive means all the time. In the presence and absence of lactose, there's constitutive transcription of the structural gene. And I'll throw throw up a figure from the book in a second, I think. Right? In the presence of lactose, there's transcription. The repressor doesn't matter. In the absence of lactose, it's usually binding. If it cannot do that because of a mutation that renders it non-functional, there will always be transcription. So in our before and after, they don't look like on and off, but the wild type system is always on. All the structural genes are always being transcribed. So constitutive transcription. So what other things do we need to know about mutant alleles usually? In a haploid case, this wouldn't be important, but this would be really important in a diploid case. I'm going to give you a problem, but you need a piece of information to solve it. Where they're located, I think that, yeah, that's important to think about. But what's something specific when we now move to 2 copies that we didn't have to worry about when we had 1 copy? Yeah, so in chat, someone put dominance. You need to know the dominance relationship between mutant alleles and wild type alleles. Often non-functional, we say, are recessive, but you need to know that explicitly, right? So LAC-I, the wild type allele, is dominant to this non-functional mutant allele. This is not always going to be the case that all mutants are recessive, so we should think about that explicitly. So here's our partial diploid. So we've separated now the mutant and wild type alleles of LAC-I, the regulator, and LAC-Z, the structural gene. And the question is, what are they producing in terms of beta-galactosidase? So we're thinking about beta-galactosidase because that's the structural gene encoded by LAC-Z. So I didn't write the question, but it's in top half. So what's the phenotype of this mutant? About 30 people have answered. It's a 2-to-1 ratio B to A. Does anyone want to start commenting on how they thought about this problem? Okay, so it's someone put it in the chat, a single functional beta-galactosidase is sufficient. Yes, so, but how is it being, so I'll just do one thought exercise, and then you tell me where I'm going wrong. So we have a mutant LAC-Z, so this is never going to produce anything, right? But then we have our functional LAC-Z is hooked up, it's in linkage with this non-functional repressor. So how is it that we're going to produce any beta-galactosidase if we have this mutant repressor? So I agree, there's the potential to express wild type levels where we have this mutant. Why, if we are going to express wild type levels of this gene, how is that happening? the chat, someone said the wild type can regulate. Yeah. So this can bind to this promoter, right? We didn't write the full genotype. We care about just the genotype at the partial diploid part, but there is still a promoter, there still is an operator. So that's called transregulation. So we barely covered that a couple lectures ago. So LAC-I can still regulate both chromosomes, whatever is on the plasmid or the endogenous bacterial chromosome. One copy of LAC-I wild type, and it's expressed in the cytoplasm, and then it can interact with the DNA. So the phenotype, your choices of the phenotype, where, well, there's a non-functional, that's true, but that's what I crossed out. It doesn't really matter because there is a functional that can be regulated by LAC-I. And then, so wild type production, that has a lot of things in there, right? It means that it's still going to operate, the system is going to operate similar to wild type. In the absence of lactose, no transcription. In the presence of lactose, transcription, because of that wild type LAC-I. So the best answer here is wild type production. If the wild type in the amount doesn't matter. It's wild type in the amount, but also the before after. This system is still negative reducible, even though you have two mutations on two different chromosomes. So this is what it looks like in your book. So I'm not going to walk through all of it, but I would become familiar with how the book is represented in these. There's a draw to learn this week where you have to pick a mutant and draw it like this. Hopefully it will help you think about what's happening. So we briefly covered cis and trans a little bit, a couple lectures ago. And we use those terms because they're historical, but I think it's easier to remember that cis is something that's on the same piece of DNA. This is going to be important for the other mutations we talk about. So on the same chromosome. And they don't have to necessarily be close. In eukaryotic systems, we have things that are cis-regulated that are thousands of megabases away. We'll see, oftentimes in eukaryotic regulation, things fold upon each other, but they can still only physically interact with things on the same chromosome. So something that is regulated in cis is on the same chromosome. Something that's regulated in trans is something that I think of it as like it's a common factor of the cytoplasm. So trans can move around the cytoplasm. Bacteria don't have nuclear membranes, so that's why I'm saying cytoplasm. So here you have this is the full genotype that we just wrote out, right? We wrote LAC plus and LAC Z minus, right? So this is the genotype of this strand of DNA. Oops, I should not have rewritten my slash. And this is our other genotype, LAC-I minus, LAC Z plus. And then because those repressors are just transcribed, translated, and are in the cytoplasm, they can interact with either piece of DNA. So we maintain negative repressible. So we said previously that the wild type allele of LAC-I plus is dominant to LAC-I minus. You could add another term to that if you wanted. You could say it's trans-dominant. That's going to be important because we'll see other mutations that are cis. So this is just zooming in on the dominant part. Oh, this is a different mutant. Okay. So here's a genotype, a different genotype. So I don't know if you can tell here, this is written as LAC-IS. So how would you write the genotype of this one? LAC-IS, and it has a functional LAC-Z. So I can put a plus. And then on the other one, there's a normal wild type LAC-I and a functional LAC-Z. So in this case, there's a dominant suppressor, which is also trans-dominant, but it's a suppressor. So this system would be constitutively off. So lac-I S is dominant to lac-I is dominant to this other mutant thing. So I don't have a question about this one, but it's a really important suppressor to think about. It doesn't matter about the genotype anywhere else in the system. If you have a dominant suppressor, you'll never see transcription. Is it kind of scary to see that in this example? Okay, so we started kind of left to right. We started with mutations in the regulator. We're going to move to mutations in the promoter and operator. So the operator here is not a unique chunk of DNA, but it's the overlapping chunk that's regulated are going to interact between the promoter and the first few bases of LAC-Z. So this is the mutation we'll mostly deal with here. So it's LACO-C, and the C stands for constitutive. So what does that mean again?
Always expressed?
Yeah. You can say constitutively on or off. I think the book typically is using constitutive to mean always on. So what other information do we need to know about this mutant? We need to know about its dominance. This dominance relationship actually isn't as important. Does anyone have a guess? Take a guess why this dominance isn't really that important? Or how are we going to qualify this dominance? Cis-actin. So whereas the regulator was common to a cytoplasm and can interact with any DNA, this is dominant, but that really doesn't matter because it's cis-acting. Whatever chunk of DNA this LAC-OC is on is going to express structural genes constitutively, but only on the same strand of DNA. It has no effect on the homologous sequence. What's the phenotype of this mutant? Should be the next question. So on one chromosome we have both chromosomes we have wild type regulators, LAC-I plus. One chromosome we have LAC-O, the wild type with a non-functional Z allele. On the other chromosome we have the constitutive LAC-O with a functional Z allele. So what is the expression going to look like in this mutant? Maybe the options are not the best, but we'll see what happens. So similar to last time, I think we can rule out A. It is true, there is a non-functional beta-galactosidase, but expression will happen. So don't get thrown off by just seeing a minus and thinking that the system is kind of a dead end. You have to look at all the components of the system that are interactive in this part of the protein. So ruling that out, about 60 people have answered, and it's even split B and C. And this might be my fault for not making this option more specific. But does anyone want to talk about B or C? So B is wild type expression levels, C is lower levels. Any thoughts? What if I change B to say wild type expression all the time? That would be my preferred answer, right? If I'm looking at this problem, I see a non-functional lag Z. So in the absence of lactose, let's just look at the left-hand side. In the absence of lactose, no beta-galactosidase. In the presence of lactose, no beta-galactosidase. That's on the left-hand side. So this is no beta-galactosidase. Ever, right? For the non-functional mutant, never expressed correctly. But here, there's always beta-galactosidase. Bacterial are normally haploid, so I would say the best answer here is wild type expression in the sense of the level. Bacterial are normally haploid, you're going to express as much beta-galactosidase as you need, but it's going to be all the time. So if that threw you off, I apologize for that. B, you should say wild type expression all the time. But it's definitely not going to be lower. Whatever's happening on this other side doesn't really matter, as long as you're getting some expression of LAC-Z, which we are, from this LACO-constitutive unit. So that's what's shown here, right? So they're both making the same in the top, they're both making the same repressor. In the absence of lactose, we get beta-galactosidase. In the presence, we get beta-galactosidase. So that one kind of half of the partial diploid is never contributing to functional beta-galactosidase, but it doesn't matter because the constitutive operator mutant is physically linked to the wild-type LAC-Z. So there's a bunch of moving parts. So what's the order of operation? Where are you going to look? I would look in the order that we're presenting them. I would first look at the regulator, so that can act trans, and then I would look at my cis mutations. So mutations of the promoter and the operator, and then structural beats. So I would look at structural beats last if I was to come up with an algorithm to how to solve these problems. So here's the flip side, right? So this is the opposite genotype. So now we flip the constitutive, right? So this part is the part that changed. So the system would look like a normal bacterial system. You don't really ever see the contribution of that LACO mutation because in the absence of lactose, nothing is being transcribed as functional, only in the presence. So just by changing the linkage relationship of this constitutive mutant, we get a completely different phenotype, right? So I'll go back. So here, it was hooked up to a wild type allele, so it's always producing. Here, it's physically linked to the mutant Z allele. It's never producing functional, but the other half is, right? This is the wild type. This is like a wild type complement, so it's going to be negative and neutral. Any questions on these? So the last place we haven't looked is at the promoter. So this is another mutation that's cis-active. The promoter is what's recruiting RNA polymerase to a specific DNA locus. So it can't interact with other chunks of DNA. It only interacts with the structural genes and the operators that are downstream of this promoter. That's how we're defining the cis-active. So what about this mutant? This should be the next topic question. I'll cut that question, we'll just talk about it. Any thoughts on this one? I don't have a before and after picture either, so we can just talk about it. Right, is that your answer? So we have one thought of lower production. Does anyone agree with that? Feel more strongly? Can you make more stronger statements? Let's do half and half. Let's take on this right side. Is this going to produce? What's this going to produce? Yeah, non-functional. The promoter is wild type. RNA polymerase and the presence of lactose will come, right? The regulator will be gone. RNA polymerase will come. It's going to transcribe this gene, but it's going to produce non-functional beta-galactosidase. What about on the left-hand side? No what? I think I heard someone whisper it.
No promoter.
No transcription. So in this system, we're never going to see beta-galactosidase expressed. We're either going to get a non-functional protein or no transcription at all. This left-hand side, if the promoter is mutated and RNA polymerase cannot come, we're seeing no transcription. So the phenotype of this mutant would be you never see beta-galactosidase. Or another way to say it is you could say it fails to produce functional beta-galactosidase if you want to take into account that non-functional. I'm fine with you describing the system either way, as long as you can kind of rationalize your answer. So here's a table that kind of, I think this is from your book. It's trying to get you to think about the consequences of these mutations kind of in a comparative way. The structural genes are only affecting that gene. Right? All the other genes will be transcribed, the proteins, they work together to metabolize lactose. But if you're just measuring expression of that protein, a structural gene mutation is only affecting that gene. And it really is only going to do it if it's an amino acid substitution, right? So we didn't really talk about different mutations, but you saw them. You saw that some mutations in a codon, the amino acid remains unchanged, right? Usually if it's in what, the third position, there's no change. If you mutate the first or second position of a codon, you get a new amino acid. So the only types of amino acid substitutions or mutations are going to be amino acid substitutions. They affect a single gene. The regulator mutation is going to affect transcription of all genes in the system because of that trans mechanism, right? It doesn't matter which chunk of DNA, which chromosome it's on, it's going to be transcribed and translated independently, and those proteins can go anywhere in the cytoplasm. Operator and promoter mutations have to be in cis. They're going to affect anything downstream of them on the same chromosome. So the reason we don't talk about structural genes as being trans or cis or trans, because their effects are completely localized to a single gene. There's no other, we only think about cis and trans when we think about the effect of one gene on other genes. Any any questions about that? Okay, so for the rest of the lecture, we're going to briefly touch on positive control, just to give you an example, and then trip operon, just to give you an example. So you don't need to memorize anything about the trip operon in the detail of the slack operon, but I think it's really important to think about how it's being regulated, because that will foreshadow how we're going to think about eukaryotic genes being regulated. So what's positive control? Can someone remind us what positive control is? Remind me, I don't remember what positive control is.
That's when the regulatory protein is an activator.
Sure, yeah. So we have a protein, it's going to be an activator, and it's going to increase transcription. Right?
Correct.
So you might think that this superficially looks like a negative inducible system where there's no transcription and then there is, right, like off, on. This will be off, on, but it's not due to a repressor, it's due to an activator. So there was nothing physically inhibiting transcription here in this scenario, but it requires a new protein. That's if you define kind of positive control in general. For the lac operon, we're going to see a very specific mechanism. But I would define positive control as a protein. It means a protein that's an activator that's going to increase or turn on transcription. So specifically in the lac operon, it's going to be really important to think about the levels of glucose. And then another small molecule called AMP. So the activated protein that we're going to be dealing with is it's called catabolite activator protein, so CAP. And there's a small molecule that it's going to interact with, which is AMP, cyclic AMP. And what's interesting about this system, right, the end product of the lac operon is glucose. So these two small molecules are inversely related. So if you have higher amounts of glucose, you have lower amounts of AMP. And the reciprocal is true. So this is kind of like a feedback loop where the metabolite is going to suppress kind of transcription in a different way. So I don't have many details about AMP, just if people really care. So it's adenosine. And then I said it's inversely proportional to glucose. So you don't need to know what CAMP is, but if you're wondering, cyclic refers to the structure, right? AMP comes from adenosine monophosphate. So this is a picture of how positive control is going. So when glucose is low, CAMP is high, it's floating around in the cell. We have our CAP activator proteins going to interact and bind upstream. So this is really important. CAP is binding upstream of the promoter. Regulator proteins get in the way of RNA polymerase, right? The regulator we saw previously is at the operator. RNA polymerase can maybe come, but it physically can't go anywhere. This is not what is happening. This is binding upstream and recruiting RNA polymerase. So the physical orientation is very different. The book doesn't draw LAC-I. LAC-I is not playing the role, right? LAC-I is playing the role in negative induction. This is no longer negative induction. This is positive regulation. So if glucose is low and lactose is present, the cell wants to make, right, metabolize lactose into glucose. So it can do this through a different signal. It can do this through positive regulation. The other similarity, though, is that it takes kind of a small molecule to activate a protein, right? CAMP is going to activate the CAP protein. So what happens now if there's a lot of glucose? So we're not talking about why they're inversely related. We're just going to use that as kind of a tool to think about it. So now when glucose is high, CAMP is low, there's not enough material, there's not enough small molecule to activate CAP, so there's no positive control. RNA polymerase isn't doing a great job binding to DNA by itself. It needs kind of proteins to help it. CAP is one of those. So again, it has superficial similarities to how the system is being controlled, right? It's kind of off and on, or whichever way you're looking at it. But it's not due to interactions at the operon upstream even of the promoter. Any questions about that? So you don't have to memorize a lot about this, but I would just think about it as kind of, there's a lot of parallel. We need to activate a protein using a small molecule. We need to interact with an operon, but how we're interacting with the operon is very different. So why do we want to think about this? Like, why is this something interesting to think about?
Diabetes.
Because this complex produces a sharp bend in DNA. Right? It changes, not the structure, but it changes kind of like the physical orientation of the DNA to bring things closer together. Right? RNA polymerase can bind in a different way to this bent configuration than it could have been in kind of the normal native DNA configuration. Does this picture look like anything else we've seen before?
Yeah, also looks like.
Think back to a couple lectures.
Looks like a transfer RNA.
Where else did we see sharp bends in DNA? This is exactly how in eukaryotic systems we bring together transcription factors and enhancers. Transcription factors are interacting with DNA, causing a physical change or a bend to bring kind of proteins that are going to increase transcription closer to the promoter. Transcription factors and enhancers can be thousands of base pairs, millions of base pairs away from a promoter. They physically interact through this DNA bending. So a lot of genetics classes will go really into depth on this mechanism. I just want you to see the superficial similarity. The reason we care about positive control in E. coli is not because it's inherently interesting, it's because it can teach us about how eukaryotic genes are regulated due to this physical similarity of the DNA bending. Does that make sense?
Yeah.
So if you go to old lecture slides that we did on transcription, and you look up transcription factors, or in your book, look up transcription factors, you'll see pictures identical to this, just with lots of more proteins. Okay, to recap what we just learned, let's see how much sink in. What's the effect of a high level of glucose on this last operon? So with about 50 people answered so far, I'm getting an even split between B and D. So does anyone want to talk about either answer? So B is little transcription taking place, and D is that we don't really know because we need to know about lactose. Yeah, so someone put in chat, we didn't talk about lactose for positive control. That's a good thing. Yeah, and then someone put it's that negative inverse relationship with CAM. So those are two ways I think, so the best answer here is B. And there's two ways you can think about it. Positive control, we didn't think about lactose at all, so we can roll out D. That leaves us with kind of no effect, increase in transcription, or no transcription, right? So A and B are kind of opposites in signs. C is just nothing's really happening due to this. So I think you have to think about that inverse relationship. So if glucose is high, then the CAMP must be low. This is the small molecule required to activate CAP. So if CAMP is low, you can't activate CAP, little transcription sequence. Questions on that one? I'm less interested about positive control than I am thinking about partial diploidy. So you'll see in discussion tomorrow, we're not going to do problems on positive control, we're going to do butyl. What we are going to do, though, in discussion is take a whole new operon and just first principles, and we say anything about the regulation based on data. And so that's what we're going to do a little bit here, just to get you, we've been talking about lack. Let's do a whole new operon. See how our logic that we're building can transition to a new operon. So we're not going to go into much depth on TRIP at all. That's not the point. The point is, take the tools you've been learning about and apply it to a whole new operon. So what do we need in an operon?
Promoter.
Yep, we need a promoter, so TRIP has its own promoter. Yep. What other parts do we need in an operon?
Structural genes.
What puts the op in operon? Yeah, operator. So this is your operon. And then we need a regulator somewhere else in the genome. There are more structural genes in the TRIP operon than the lac operon. That's not really important. What's important is that it's metabolizing orosmate to tryptophan. So tryptophan is a small molecule that the bacteria want to produce. It's also going to be our regulatory, our small molecule that's going to interact with our regulators. So I'm going to tell you something about this molecule. So tryptophan is a co-repressor. So just with this information, that tryptophan is a co-repressor, you should be able to answer the next top-back question. The question is, in the TRIP operon, what happens to the repressor in the presence of tryptophan? So just knowing that tryptophan is a co-repressor should give you enough information to answer this. And this came up in a question last lecture too. You have to think about the perspective, like what protein am I asking you about? So I'm asking you about the TRIP repressor, right? So not necessarily the whole operon, unless it helps you answer the question, but this repressor protein in general. So if tryptophan is a co-repressor, how is the TRIP operon regulated? You guys know how to play charades?
2 words, 1 syllable.
I'm not saying I'm good at charades.
I'm trying to figure out what this. Connecting?
Negative, yes, you got it. Second word I don't have an action for. What's the second word we're going to add?
You said 2 words, 1 syllable.
Repressible.
Negative. Oh.
If you have a co-repressor, the system has to be regulated in a negative repressible fashion. if there's a small molecule that's going to bind to a protein to repress, that has negative action, right? The negative is the regulator is going to bind and turn something off. The before and after is going to give us inducible or repressible, but we defined it in the name, right? Allolactose is the inducer and the other operon. Here we have a co-repressor. So in a negative repressible system, what happens in the presence of this co-repressor? So the best answer here is A, right?
Okay.
We were on, and now we want to turn off. So tryptophan is going to bind to the tryp repressor,
So it goes to the whole operator, not the regulator.
and it's going to bind to the operator and repress transcript. So we started here, I gave you information about the co-repressor, then that helped us determine the way that we're going to talk about this. Negative repressible, then that will help you answer the question. So this would be the case of negative inducible, right? B describes an operon that's negative inducible.
Mm-hmm.
C is a tricky one. It binds to the regulator gene and represses transcription. Yes, I prefer A because it's getting at the operator, right? That regulator gene cannot do anything in and of itself. It has to physically interact with the operon. So that's why A and C are similar. That's why I prefer A. D is also more similar to something you've seen under negative inducible conditions. So the best answer here is A. So this is what the tryp operon looks like. When tryptophan is low, you have an inactive regulator. So this is the other thing not to get caught up on, that binding becomes inactive, right? Whether a protein binds to a small molecule, it's not always going to be a one, like that's not always going to make the protein active or inactive. It really depends on the context of that protein. So we have an inactive regulator that cannot bind to the operator. Transcription is happening. Now when tryptophan levels are high, you've kind of metabolized enough. Cellular efficiency would select for a system that has a kind of a feedback loop, right? Tryptophan is high. There's enough of it circulating in the cell that it's going to turn this inactive regulator into an active one that can bind to the operator. So this is what gives us negative, right? That repressor is binding and inhibiting transcription, and what gives us repressible is on to off. So the other really interesting thing about this, that your book spends a lot of time on, that we're not going to spend any time on, is called attenuation. And the only thing I want to mention about attenuation, so what is attenuation? Attenuation is when transcription is stopped due to translation. And well, how does that work, right? We've always been talking about kind of this linear progress in the central dogma, right? Transcription, translation. Transcription, translation. In eukaryotic cells, they are separated in space and time by the nuclear membrane. In prokaryotic cells, they are not. Right as transcription is happening, right as there's free mRNA, a ribosome is immediately there. So in the tryp operon, there are certain chunks of the mRNA that can make loose. This causes RNA polymerase to stall. Your ribosome is chugging along, it can actually bonk RNA polymerase off. So you don't have to memorize any of that, you're not responsible for it, but it's a really cool mechanism that these are dynamic processes. It's not happening kind of one-to-one, they're happening simultaneously in these cells. People can take advantage of technology like that to do lots of different types of biotechnology, medicine, like you can construct things so that you can stop translation and transcription through altering DNA sequence. So that's all I'm going to say about attenuation. So if you're interested in that mechanism, you can read more about it in the book, and you won't be responsible for it in the exam. Okay, that's my last slide. Even with all my pencil fumbling, we finished early. Make sure you come to discussion. This unit that we're in the sense of like material that's going to be on exam 3, we don't have a quiz, so it's all fair game, right? Previously there was a pattern. If you had a quiz on something, you didn't see it on an exam. We don't have enough time to do a quiz, so you're going to see all of this on the exam. So make sure to come to discussion, hear solution strategies. I can stay for a couple minutes to answer the questions.